Every animal you can think of, from a massive blue whale to a tiny fruit fly, started from just a few common blueprints. Today, we're going to rewind the clock and go way back to the very beginning. We're going to look at the first, simplest designs that laid the groundwork for all the incredible complexity we see in the animal kingdom today.
Okay, I know this chart looks a little intense at first glance, but trust me, it's our map for this whole journey. It shows how scientists sort every animal on Earth by asking just a few fundamental questions. First, are an animal's cells just a loose collection, or are they organized into specialized teams, like tissues and organs? Second, what's its body plan? Is it kind of random, like a sponge, or symmetrical, like a pizza? And third, does it have an internal body cavity? The answers to these questions are what separate all the major branches you see on the tree of life.
So, here's our game plan. We'll start with the simplest of the simple, the sponges, also known as Porifera. Then we're going to see a major leap in complexity with the stinging Cnidaria and the absolutely beautiful glowing Ctenophora. After that, we'll see a revolutionary new body plan with flatworms and roundworms, before we wrap it all up and look back at the incredible evolutionary ladder we've just climbed.
All right, let's dive right in with our first stop, the Phylum-Prifera, which you probably know better as sponges. Now, these might seem more like plants or even just cool-looking rocks, but they are absolutely animals. In fact, they are the most basic blueprint for multicellular life, giving us a window into what the very first animal communities might have looked like.
Sponges really do come in all sorts of shapes and sizes, from simple tubes to these big blobby masses. And you'll notice something right away. None of them have a distinct front or back, or a left or right side. They're what we call asymmetrical. This lack of a defined shape is a hallmark of their very simple, yet incredibly successful design.
So what makes a sponge so, well, primitive? It really all comes down to this, the cellular level of organization. Think of it like a group project where everyone is pitching in, but there are no assigned roles or specialized teams. The cells work together, sure, but they haven't formed dedicated groups like tissues or organs. It's really the simplest way you can build a multicellular animal.
The absolute key to a sponge's survival is its incredible internal plumbing. It's basically a living filter. Water is constantly being pulled in through thousands of tiny little pores called ostia. That water then flows into a big central cavity and finally exits through a large opening at the top called the osculum. This simple but brilliant system is how they eat, breathe, and, take out the trash.
And what powers this whole system? Well, it's these amazing little cells called choanocytes, or collar cells. They line all those internal canals, and each one has a tiny little tail, a flagellum, that whips back and forth to create the water current. Then their special color traps tiny food particles from the water. They are the engine and the entire digestive system all rolled into one. Pretty amazing.
Okay, get ready for a major leap forward on the evolutionary ladder. It's time to meet the phylum Cnidaria. This group includes some really familiar creatures like jellyfish, sea anemones, and corals. And let me tell you, they are a huge step up in complexity from the sponges. So right away, we're seeing our first major design upgrade, symmetry. Cnidarians have what's called radial symmetry, which means their bodies are organized around a central point, kind of like the spokes of a wheel or slices of a pizza. This group also has two main lifestyles. You've got the floating bell-shaped medusa form, which we all know as jellyfish, and the stationary polyp form, like a sea anemone, which tends to stay put. This is a huge deal. Unlike sponges, Cnidarian cells are now organized into true tissues. They're also what we call diploblastic. Now, just think of it like this. You're building something with two layers of material instead of one. They have an outer layer, the ectoderm, which becomes their skin, and an inner layer, the endoderm, which becomes their gut. This two-layer plan is a really foundational step for building a more complex animal.
And now we get to the Cnidarian's signature feature, their secret weapon, the very thing that gives them their name. They have these incredibly specialized stinging cells called clinidoblasts. And you can see a diagram of one right here. These tiny biological harpoons are the reason a jellyfish stings. So these cnidoblasts, which are all over their tentacles and body, contain this little spring-loaded stinging capsule called a nematocyst. When something triggers it, it fires a tiny venomous thread. It's an absolutely incredible adaptation that they use for everything from anchoring themselves to rocks, to defending against predators, to capturing their next meal. And this just lays it out perfectly. You've got two totally different lifestyles in this one group.
You have the polyp form, which is stationary and cylindrical, just like a sea anemone. And then you have the medusa form, which is free-swimming and umbrella-shaped, like your classic jellyfish. Some species even get to be both, alternating between these two forms during their life cycle. Next up, a closely related but totally distinct group, the Ctenophores. You might know them as sea walnuts or comb jellies. They share some features with Cnidarians, like that radial symmetry and having tissues, but they also have some spectacular and unique features that are all their own. Here's a classic example, Pleurobrachia. Now, at first glance, you might think, hey, that looks a lot like a jellyfish. And you wouldn't be wrong. They're both marine drifters with a pretty simple body plan. But if you look closer, you'll see that Ctenophores have their own really unique and beautiful way of getting around. Their defining feature, the thing that makes them so unique, are the 8 external rows of shimmering comb plates. These are made of tiny little hairs or cilia, and the coordinated beating of these cilia is how they move through the water, creating this beautiful rainbow effect. And, as if that weren't cool enough, many Ctenophores are bioluminescent. That means they can produce their own light, creating these stunning, silent light shows in the deep ocean. Okay, so everything we've looked at so far has been either asymmetrical or radially symmetrical, kind of random or built like a circle. But now, we're at a huge fork in the road of animal evolution. The next groups we're going to look at introduce a body plan that will literally change everything: bilateral symmetry. First up are the Platyhelminthes, or, flatworms. As their name pretty clearly suggests, they have a distinctly flattened body. And while some of them are free-living, many, like the tapeworm and liver fluke you see here, are endoparasites, which means they live inside other animals. And here it is. This is the real game changer. For the very first time, we see bilateral symmetry, a distinct left and a right side, a front and a back. This simple change allows for a head to develop at one end, which is the first step towards having a brain and actively seeking things out. They're also triploblastic, meaning they have a new third embryonic layer called the mesoderm. This middle layer is the raw material for building muscles and complex organs. The only thing they're still missing? An internal body cavity. They're basically solid all the way through. Now let's check out the Aschelminthes, or roundworms. You can see the difference right away. Their body is circular if you look at it in a cross-section. This group, which includes creatures like Ascaris, has taken another step up the ladder to the organ system level of body organization. And here is the next big innovation. Unlike those solid flatworms, roundworms actually have a fluid-filled body cavity. It's called a pseudocoelom, which basically means false cavity, because it's not fully lined by that new mesoderm layer. You can think of it as an unlined pouch. This simple space acts like a hydrostatic skeleton, kind of like an internal water balloon, giving the worm's muscles something to push against, and that allows for much more efficient movement. All right, we have covered a ton of ground. We've gone from simple collections of cells all the way to animals with distinct organ systems and body cavities. So let's just take a second and recap the key advancements we've seen on our journey so far. This table just lays it all out so perfectly. You can really see the step-by-step progression. We went from loose cells in Prifera to organized tissues in Cnidaria. We saw the shift from no symmetry to radial symmetry, and then that revolutionary jump to bilateral symmetry in the flatworms. And we watched as animals went from being solid masses to developing that first simple body cavity in the roundworms. Each row here really represents a major evolutionary breakthrough. We've seen an amazing progression, this step-by-step increase in complexity, from simple sponges to the more advanced roundworms. Each step, tissues, symmetry, a third germ layer, a body cavity, it opened up a whole new world of possibilities for animal life. So this leads to the final question. What was the next big breakthrough? What innovation allowed for the explosion of diversity that led to insects, fish, and eventually us? Something to think about. Thanks for joining me on this journey.